This talk is called Overcoming DNA Stochastic Effects, and thank you all for coming. Next slide. Forensic scientists need to cope with uncertainty in many ways. Though that our DNA data are reproducible, they do exhibit stochastic variation. Now, probability models can capture these stochastic effects, and when we infer genotypes from such uncertain data, we obtain probability distributions, as with the CPI method with mixtures. We have two requirements. Science expects us to account for stochastic effects, but the law also expects us to testify within our certainty. Fortunately, we have the likelihood ratio, which meets both the demands of science and the law. The likelihood ratio expresses the data and genotype uncertainty in a way that can be used in court. Next slide. The key source of stochastic effects in DNA forensics is the polymerase chain reaction. PCR is a random process because every cycle, every strand of DNA can either be copied with some probability, say 80%, or not copied with some probability, 20%. This is like flipping a biased coin where there's a probability of success or not success. The result is that PCR is a random branching process, and it does not have 100% efficient replication. Next slide. Let's see how this plays out with an STR peak. An STR peak is actually a random variable. If we do a computer simulation with a probability of success in that coin flip experiment at 80% and run it a few times for a number of copies, what we'll see is that in one amplification, we may achieve some copying rate that's less than doubling each time and get 1,300 copies. And yet, starting from exactly the same materials on another amplification, we again keep increasing by under a factor of 2 randomly each cycle. We end up with 2,900 copies. So that's over a factor of 2. Next slide. The STR peak height measurement in our DNA sequencer reflects that probability distribution of what the random variation is of the data stochastic effects. As a probability distribution, theory shows that it looks very much like a bell curve or a normal distribution. And it has a mean, you can see in the number of copies or in the RFU, and a standard deviation. In other words, we do not expect to get the same peak height every time. That's science. Next slide. It's useful to describe the relative peak certainty as the coefficient of variation. The coefficient of variation is the width of the bell curve divided by the center of the bell curve. So it's the width over the center. For example, the standard measurement will be a standard deviation over the mean value. Here we see a standard deviation of 12 million units with a mean of 85 million units, and the coefficient of variation of 12 over 85 is 14%. In order to obtain greater peak certainty, next slide, we need 4 times the amount of DNA. That would give us 4 times the average peak height to achieve twice the peak certainty as measured in coefficient of variation. Notice here, we increase the mean 4 times up to 350 million units. The standard deviation doubled from what we had before to 25, but that ratio of doubling of the numerator but quadrupling of the denominator halves the coefficient of variation, and so tightens our certainty around that peak. If you see the ratio of the red width to the orange extent. And the way you see it is that the peak is much more concentrated around the region of the peak height with a lot more white space of where it can't be underneath it. Whereas in the previous slide, there was the peak, the probability distribution of the peak, occupied much more relative area than did with far less white space. That's, by the way, it's the easiest way to see it. Here it's taking up half, and here it's taking up a quarter. It's the white space. Next slide. So this shows that STR peaks are random variables. When we do an STR experiment, we're sampling from those random variables, and we can see at different sizes different amounts of RFU, and what these bands represent is the amount of bell curve variation that depends both on the peak height and on the certainty of the experiment. More confident experiments will have more concentration around the peaks, and as you can see, the amount of variation scales with the peak height. The coefficient of variation decreases as the relative variation with taller peaks. We have more relative confidence in it. Next slide. Now, to interpret quantitative STR data, some groups use a qualitative threshold. And this is what they do. You don't want to cast any light. So suppose we have a threshold of 50 RFU applied to our quantitative data. We should probably introduce the axes. Here's the size and base pair. Here's the RFU. Here's the quantitative data. Now we apply the threshold. Over the threshold, the peaks are treated as allele events. So we would see alleles at 28, 29, 30, and 32.2. However, under threshold, we imagine that those alleles do not exist. And so we end up with a list of the included alleles that represent peak events over threshold. These are all or none events. There's no peak magnitude left anymore. Either they're present or they're absent. However, because STR peaks are random variables, thresholds introduce error. If we set a threshold of 100 and we observe a peak that's under 50 for this event, then we would give one answer, and if we state that we observe a peak that's sampled over 50, we would give another answer. So there is a true answer as to whether or not these events represent alleles, but what a threshold does is that it introduces the possibility that if this is an event, you have a threshold of 200, if you sample above it, then you're correct, but if you sample below it, then as you see from the probability distribution, you may have a 50% chance that you're wrong. Now, those probability distributions did not have any error until you drew a threshold. The result of this error, next slide, is false allele exclusions. So this is a study that we did. In fact, Kara Spencer, along with my compatriots, Jessica Staub, as she was called at the time, and Erin, she was married, Churro, at Cybergenetics, we, in triplicate, examined a host of data that's described in our 2009 paper, An Information Gap. And what we did is we, in triplicate, applied thresholds to synthesized mixtures that were made from a pair of NIST samples that were provided to us by NIST. And we knew what the correct alleles were. In this study, there were two pair of individuals at 5 different mixed proportions, 90-10, 70-30, 50-50, 30-70, and 10-90, in 4 different dilutions. 1 nanogram, half a nanogram, quarter of a nanogram, and an eighth of a nanogram. So what we did is we simply observed at 3 different thresholds of 50, 100, and 200 RFU what alleles we would call based on those thresholds. Then since we knew what the answers were, we were able to determine the false positive and the false negative rate. There were 4 false positives observed in the study. So I'll show you now the results on the false negatives. These are reported in the number of falsely excluded alleles per locus. What we see with the 50-50 mixture is that at 1,000 or 500 picograms with a threshold of 50, we're not falsely excluding any alleles. However, as the amount of DNA gets less, at 250 or 125 picograms, we're now beginning to exclude alleles. How many? Well, it ranges from maybe 10% to 60%. 60% of alleles per locus, on average, would be wrongly excluded. And you can imagine why that would be true as the uncertainty of the peaks get lower and the peak heights get lower and lower. So with 50 RFU, it has to do with how low the peak heights are. At a 30-70 mixture, again at 1 nanogram, I'm just describing what I'm seeing. We don't have any observe any false negatives. However, even at half a nanogram, the false error rate has gone up.
It looks like it changed the axes in Keynote.
It did.
You just won't save it. That way there's no harm.
That's fine. I'm on that.
It'll be okay.
Fine. I'm just looking at it. I'm trying to make up what the numbers are. So about 30% for 250 picograms. And with the lower peaks that we observe at 125 picograms, the error rate is up to about 100%. That is, on average, there will be one miscall of an allele that is falsely excluded at each locus. When we have more extreme mixtures, like 90-10 mixtures, we then begin to see an even higher error rate as we go from 30% to 50% to 100% to 150% with decreasing peak heights that are associated with the decreasing amounts of DNA. Now, the 2010 SWGDAM Guidelines recommend doing calibration experiments on data that have the effect of raising the stochastic threshold. That means that the peaks that we saw before at a threshold of, say, of 150 or 200, we would no longer see in these experiments. When every peak is under threshold, all the data disappears, and we're left with no genotype, no match score, nothing left to say. I'd like you to actually say those phrases with feeling, because they were written as poetry and haikus, not merely as words on a slide in this particular instance. Thank you. Next slide. Just when every peak, you can see it has a certain rate. This produces even higher false exclusion rates, as we see here. With a threshold of 200 RFU, we're in the range where many labs have moved to now, the false negative rate of alleles per locus at different DNA mixture ratios has visibly gone up. Even with 50-50s, where all the peaks are roughly of the same tall height, at 1 nanogram, there's a lower rate of maybe 10%. At 500 nanograms, though, we're up to 60%. At a quarter of a nanogram, where the peak heights become even low and more stochastic-y, the error rate is now over 100%. At an eighth of a nanogram, the peaks are so far below threshold that the error rate is at 150%. That is 1.5 excluded alleles per locus on average. With the DNA mixture ratio of 30-70 or 70-30, the rates go up now, ranging from 40% up to 160%. And with 10-90 mixtures, the exclusion rates from the known two contributors, the error rate, is now going up in the range of 100% to 170%. So while the examiner may have greater confidence as to what's left, typically in science we don't see false error rates of over 100% when we examine our data. Now, fortunately, the SWGDAM 2010 guidelines come to our rescue. They say if you don't use stochastic thresholds, then you can use a probabilistic genotype approach, where you make better use of the quantitative data. You have to support your criteria with empirical data internal validation. Now, whereas higher peak thresholds may discard information, probability modeling has the potential to preserve all the information that's present in the data. Next slide. What is probability modeling? In brief, there's one theorem of probability that's 250 years old that lets us address scientific uncertainty. It's the only one that we have, actually. What it says is we start with an initial probability distribution, say population frequencies of genotypes, before we see data, and then the likelihood function engages the data to update our probabilities to what we believe after we see the data. A joint likelihood lets us combine independent evidence in ways that we're about to see. So what are all these terms? Likelihood describes how well parameters explain the data. These parameters may be genotypes, mixture weights, peak certainty, stutter, relative amplification, and so on. The STR data must be explained at every peak in terms of the RFU. Now, the likelihood gives us the probability at one peak and the smallest possible level when we compare one peak, predicted peak, to an observed peak. But then, to look at all the peaks in a STR pattern at one locus, mathematically, we simply multiply together the individual peak likelihoods. This is very easy for a computer. Computers excel at arithmetic. You can go on if you'd like about adding, subtracting, and doing. So what happens to infer genotypes by Bayes' rule, or Bayes' theorem, is that all possible patterns generated in all possible ways from all varying values of parameters, all possible genotypes, values, allele pairs, all possible mixture weights, quantities of DNA, stochastic variation of the peaks, and so on. All these variables are tried out, and their patterns are compared against the data. The data here are shown with the probability of a peak, because that's really what's being compared. We don't know what the underlying DNA quantity is exactly. We only know it up to probability based on the peaks that we observe as the mean and the data uncertainty around it. Those patterns, for example, a major contributor in orange with a minor contributor in blue producing a pattern, those predicted patterns that fit the data very well at each peak. Here we see 0 is fitting 0 well. A peak height of 50 is fitting this well. Moving on, the higher orange peak is fitting. The zeros fit. The blues fit. Everything goes within the range of the data. We multiply the likelihood at each peak to get the joint likelihood of the entire pattern, and that gives us the likelihood value. Trying out all possible patterns will tell us which genotypes or other parameters have higher probability and which have lower probability. Next slide. The key point of this talk is that we can overcome stochastic effects in this way. Why? Because computers can solve, just read the slide, computers can solve for genotype probabilities and other random variables, such as the peak variation. Is it a narrow variation? Is it a wide variation? Computers can solve for that. By modeling peak variation is just one other parameter, like mixture weight or genotype, computers can overcome DNA stochastic effects. Why? Because you then know the certainty of the data. When you have very broad patterns, very broad data with very little uncertainty, those bell curves get very wide, then many patterns can fit, many genotype values are possible, and there's less certainty. However, if you know that the peak variation is very tight with a small standard deviation, very few patterns can match it, can explain that data, and therefore very few genotype allele pairs can explain it, and that concentrates the probability into the more likely genotype values based on the observed STR data. In the same way that thresholds are a very crude way of a one-size-fits-all across all the data trying to establish some control over stochastic effects, with modern statistical computation, which started about 20 years ago with the Gelfand and Smith article, if you want to be so erudite and reference it, but you don't have to. Computers can now determine the variation not across the laboratory, but for every STR peak in your data. It's a much more refined approach that only computers and probability modeling can do. And that's why you no longer need thresholds, because the computer is determining your certainty to a far greater extent than is possible by drawing one line for your entire laboratory. Next slide. True allele casework is a system that implements probabilistic genotypes. It determines peak uncertainty. It determines genotype probability distributions and thousands of other variables. Its goal is to preserve all the identification information that's present in the data. The system is over 10 years old and has been used on over 100,000 evidence samples. You can read about it more in your handout. I'd like to tell you how we use our probabilistic genotype system. Those are the exact words from SWGDAM, probabilistic genotype. In a case that we solved and testified in a year and a half ago, the Commonwealth of Pennsylvania versus Kevin Foley. For the salacious details, I refer you to other talks on our website, which are described and given on the last slide. There were 3 different match scores testified to in this case. There was a 13,000 match score using the inclusion method, which was not overwhelming since DNA was the main evidence in the case. There was an intermediate score in the millions using an obligate allele method, and 189 billion match score obtained using the true allele computer system. What we see is that inclusion methods discard information as measured by match score, whereas probability modeling preserves information as measured by match score. This is how it happens. When the computer finishes making its likelihood comparisons, we're looking at one locus here on the true allele screen, and we're asking, what is the likelihood of different allele pairs, shown on the x-axis, relative to each other? Quantitative true allele likelihood focuses the probability on the true genotype, as we can see that virtually all the dark blue of true allele is concentrated on what will ultimately match the suspect. Inclusion methods have a likelihood method that disperse the probability across incorrect allele pairs. This is because they give equal weighting to all alleles, regardless of what the quantitative data is trying to say. And here we see, for the 3 alleles, there are 6 possible allele pairs, and here's the dispersal, the diffusion of the probability across 5 wrong answers that the data really don't support very well, as opposed to concentrating probability on correct or other possible answers. Next slide. Is this limited to this case? No, not at all. In yet another, next slide, in yet another study that I, Carol Spencer, am involved in, along with my collaborators at Cybergenetics, we looked at 16 cases and compared the actual DNA match scores as recorded in the case folder on adjudicated cases with the quantitative true allele scores obtained by computer analysis of those identical cases. These results are described in a forthcoming paper in the Journal of Forensic Sciences. Here are 8 cases that are 2 unknown mixtures. Here are the 8 cases, and here is the logs of the likelihood ratio. 10 to the 5th, 10 to the 10th, 10 to the 15th, and so on. Using the quantitative true allele system, shown in blue, more information is preserved with an average score of 10 to the 13th, which is 10 trillion, on the identical data. The inclusion method, using thresholds, has an average match score of 10 to the 7th, only 10 million. The difference is 6 log units, or over a millionfold, on the same data of how much information is preserved versus discarded. Next slide. SWGDAM has another paragraph in its mixture guidelines, paragraph 3.4.3.1. You don't need to write that down. It's in your handouts that you're holding in front of you. And it talks about composite profiles, that if you have multiple amplifications, or if you have samples that come from a common source, you're allowed to establish guidelines for looking at more than one evidence item, amplification, or electropharogram jointly. Now, mathematically, the way this is done with quantitative data is the standard approach of a joint likelihood function that combines evidence. In the same way that the joint likelihood function combined the individual peaks into a pattern of one amplification, all of those peaks across multiple experiments can be combined to form a composite profile. Probability modeling here again preserves the information. Let me show you how. This is a case that cybernetics was involved in. In England, this is the Queen versus Mr. Broughton. We'll take a look at one locus here that you're seeing, VWA locus data. This was a very low template mixture. There were 3 DNA contributors. Triplicate amplification was done. You see the 3 traces. And it's with a post-PCR enhancement that increases the relative scale by about a factor of 10. Before enhancement, these peak heights in the 200 RFU range were basically invisible. They were buried into the background noise, into the baseline. After enhancement, we see the patterns, and as you can see, these patterns, which really represent peak heights down at around 25 or 30 RFU, look different from each other because there's a lot of stochastic variation. In fact, the human result was inconclusive. No match score could be put to this case at all in any meaningful way. However, true allele interpretation was done. You can throw in my favorite line if you like, because when all else fails in England and no human or computer can solve the case, people call cybernetics. You don't have to. That was more a dig at the FSS because they haven't done anything with their silly system. So, what was the improvement? Well, human inclusion produced nothing. Did I say next slide? Produced next slide. Produced nothing. The true allele computer had a score of over 1,000,000 to 1. We can see how this happened. Here's VWA again. In brown, you see the brown bars are showing the probability distribution at the allele pairs before the computer considered the evidence. In blue, after you see the evidence. So what was the gain in probability? Well, at some allele pairs, if that happened to be the suspect, there might have been a decrease. But at Mr. Broughton's allele pair, 1418, we see that the probability after seeing the data is about 6 times higher than before, which is a likelihood ratio of 6. When you multiply each of these loci together, that's how you obtain a score of over 3 million. The joint likelihood function combined the evidence so that the probability modeling could preserve the identification information. Next slide. Is this limited to this case? No, not at all. In yet another study that we've done with the New York State on 86 items of evidence, what we see in the background of light blue is true alleles match score, what the computer obtained, with a mean value, if you look at the information, the log likelihood ratio, of 10 to the 15th. The median score was a quadrillion to 1. The human scores are shown with different methods of two-person mixtures. Actually, these weren't two-person, different mixture methods from RMP, when you're not going to have time to say all this, so I wouldn't even bother. The human scores are shown in the bars. And we notice that not only are they generally a lot less, but the peak threshold methods that were used discarded any match information whatsoever 70% of the time, whereas the quantitative likelihood methods preserve the information every time. In conclusion, quantitative DNA data have stochastic effects, and with computers we can model these data using joint likelihood functions. The standard probability modeling that we use preserves DNA identification information and can statistically combine DNA evidence, whether it's the individual quantitative peaks within one locus or the different patterns across multiple DNA tests. The key point is that exact modeling of peak variation can replace the inexact thresholds in order to scientifically overcome stochastic effects. Thank you.